Welcome to BB 101 course at IIT Bombay. Professor Srivastava is going to deliver his lecture in Molecular and Cellular Biology module shortly. Please mute your microphone and be ready with pen and notepad to take notes. Thank you. Welcome to BB 101 course at IIT Bombay. Please mute your microphone and be ready with pen and notepad to take notes. Thank you.
All right, good morning, everyone. So this is a live interaction for the D1 and D2 batch, where we are going to review two topics, cell cycle from lecture 2 and metabolism from lecture 3. And for both of these, there are several topics to actually discuss. A lot of Q&A is there to cover. So this is the plan for this session. I'll first give you some of the salient features and review about the cell cycle, different concept which we have discussed already, but some of the key points I will remind you again. Uh, Lecture 2 Q&A, a lot of students have asked many questions. We will take those. Lecture 3 also, there are many questions on metabolism, but also there are some requests that, few concepts to be explained again. So those I will again review that. Some of you have actually asked, certain some questions for the quiz one. So I'll give an overview of that. And finally, we will have the today's quiz, which will be from lecture 2. All right, so this is going to be a packed session when we have several important points to cover. But let me first start with discussion about cell cycle, which we have uh, learned in the second lecture. While discussing about prokaryotic and eukaryotic cell, a concept came that how these specialized organelles like mitochondria and chloroplast, how they originated, right? And that's where this different hypothesis, which was made by scientists like Margulis, was discussed that how a eukaryotic cell might have taken any bacteria and use that bacteria as a part of its cell, and now trying to utilize that for a specialized organelle like mitochondria or chloroplast. So there are different type of, let me just go back. Oops. All right. So there are different type of bacteria which probably would have come in touch of the cell. The one which has the ability to do the oxidative phosphorylation or aerobic activity, which is required for the mitochondria to develop eventually. That bacteria, when it came in touch with the eukaryotic cell, it got engulfed and eventually developed into the mitochondria, which is having two membranes, one membrane from the prokaryote or that bacteria, other membrane comes from the eukaryote, and that's where you have different origin of these double membrane organelle, one from the eukaryote, one from the prokaryote, and especially you can pay attention to the inner membrane and outer membrane. Likewise, then probably some bacteria which has ability to do photosynthesis, uh, they got in touch with the cell, and then they got engulfed in that and became part of that as the endosymbiont. Uh, so symbiont, term actually conveys that cells are going to help each other; they are not going to harm, and that's why they're able to get shelter over there, and then they are doing some work for the cell. So in this way, now this bacteria with the photosynthetic ability became part of this eukaryotic cell and became chloroplast eventually. So other concept which we spoke about, different phases of the cell cycle, especially we have a mitotic phase when the cell division to occur, but a much longer phase which is known as interphase that prepares the cell for division to occur, and that's where there are different parts of the interphase which include G1 phase, S phase, and G2 phase. Now, we talked about different properties of each one of these phases, especially G1 phase is preparing the cell for the chromosome duplication and replication to happen. So Before S phase start replication, G1 phase prepares all the nutrients, all the conditions are favorable for doing that. Then S phase is a very crucial where all the chromosome duplication, DNA replication activities happens. Even proteins like cyclins get synthesized. And G2 phase is the last phase of the interphase when again now it ensures that there is sufficient condensed chromosome material is available for cell to proceed for division to happen in mitosis. And then mitosis of course going to divide and distribute this chromosome to daughter nuclei with a cytokinesis process which is going to divide the cytoplasm as well. So these are the important concepts there. Uh, in the mitotic phase, we have again different involvement from prophase, pro-metaphase, metaphase, anaphase, and telophase. Of course, you have a lot of questions in this area, which we'll take shortly. Uh, Essentially, the prophase is started defining that now the chromosome material has to start getting condensed, these chromatins being formed. In the pro-metaphase and metaphase, they start getting aligned to the metaphase plate. And now these chromatids, the daughter chromatids from each of these chromosome, they are aligned over there. But only in the anaphase, they started pulling apart to the two different poles. And that's where now they will be ready to divide into two different cells, and telophase and telophase makes that completely divided. Then cytokinesis with the cell furrow or the cell plate in animal or the plant cell then makes those boundaries so that now we got two daughter cells. So this is the concept which we talked in the mitosis, and briefly we looked into mitosis and comparison with meiosis. Uh, so here main concepts are that when we take these diploid cells, in the process of mitosis, those prophase, metaphase, anaphase, and telophase which we spoke, they are going to develop into these daughter cells, which is going to have the same level of chromosome, same ploidy level will be maintained. In case of meiosis, there are two different cycles happens, meiosis 1 and meiosis 2. Meiosis 1 is having some variations. Meiosis 2 works exactly like mitosis. So the interphase only happens once in this case here for the meiosis like mitosis, although one would expect that in meiosis there are two cycles, meiosis 1 and 2, so there should be two interphase, but that's not correct. It's only one interphase before meiosis 1. The key differences here is that in the both the blue and red or the parental chromosomes, there is exchange of the genetic material happens in the process known as crossing over, which is the site for the chiasmata formation known as the recombination activity. Now the homologs, which are from the both the parental chromosomes, they are going to move and get lined up in the metaphase, which is the key difference from the metaphase of the mitosis when only individual chromosomes are lined up. Here the homologs are getting lined up, and then these homologs are separating in the anaphase. Again, that is the difference. And then in the mitosis, duplication happens, and now we got two more daughter cells made after the meiosis 2. So now total we got four daughter cells. So the key differences I hope are highlighted here. Now the ploidy level from 2n to 6 remains same here in this case as well. Ploidy level in this case becomes half of the chromosome, and the number of daughter cells become 4. And these are the key activities which are different from mitosis and meiosis. In the cell cycle, there are different checkpoints we talked, and those checkpoints are present in the G1 phase, present at the end of G2 phase, present in the M phase. They are the one ensuring that the cell is actually dividing correctly, and there are different checkpoints that are regulating those activities. In the cell, to perform these processes, there's a set of proteins known as kinases. They are involved, and these kinases, especially the cyclin-dependent kinases, they are present throughout the cell. And the cyclins are the one which are produced during this particular interphase of the S phase, when the more of the cyclin gets synthesized. So now with the cyclin synthesized, getting more accumulated, with the CDK, it is coming and binding over there, which ensures now that there is sufficient cyclin present, which means indirectly that S phase chromosome duplication has happened properly, and now cell is ready for entering into the mitosis. After mitosis get completed, the CDK and the cyclin, they get degraded. Cycles get degraded and CDK is separated now. So then that is an indirect hint that now the cells are has finished this particular part of mitosis, and it's ready to move to the next interface. We talked about cloning very briefly. In the plants have more specialized potential of the cell, that they can get specialized into any kind of cell, and that is known as totipotency of the cell. And I showed this example of taking carrot and take a cell, induce that to make callus, which the different hormones like cytokinin and gibberellic acid, one could now make the root and shoot, and you can develop the full plant. We also talked about Golden's experiment on frog, which was quite interesting experiment. 1952 or so, he did that experiment, and he showed that from the a frog egg cell, when you destroy the nucleus with ultraviolet light, that becomes enucleated egg cell. And now, if you take the nucleus from the less differentiated early embryonic stage cell and put that in this particular egg cell, which is the enucleated. So now it can result into very good success of the cloning, lot of tadpoles being formed. Whereas if you take from the more advanced cells, more developed differentiated intestinal cells of the frog tadpole, those cells, if you take and take their nuclei, that nuclei after transplantation is not resulting into good success of these clones. And that was a big observation he made, and that also first time any kind of organism cloning was shown, and that's why he was quite heavily rewarded for his activities. All right, so these are just a refresher for all of you to, to give you the salient points what we have talked in the cell cycle. Then, of course, many things I'm also going to take again in the Q&A session. So I'll first take the questions from the what we have in the forum, which was already given, and then I can take some more questions from the class live here as well. So let's take a couple of points which some of you asked very nice, interesting questions, and that is really uh, show that how much effort you are putting in, and you have really spent some time to read those things, right? So one question is that what governs the M phase of the cell cycle, which is this phase, and if uh, the control centers are actually, which is the nucleolus, which is get disappeared, how the production of the CDKs are getting coordinated, right? So there are different activities involved in performing this cell cycle checkpoint and doing the mitosis. I again just refresh this concept. There are these a set of molecules which are known as kinases. Those proteins, especially the interplay of cyclins and cyclin dependent kinases, they are very crucial to trigger and coordinate these key events of the cell cycle. And that is actually giving these checkpoints of G1, G2, and M. They are giving the answer that whether it should move to the next phase or not. So again, this is a very orchestrated event which is relied based on the several set of molecule which are kinases, set of cyclins and CDKs. Again, if you are more interested to learn a little bit more detail about it, which I have not talked in the class, a part of that is also a complex being formed, which is known as mitosis promoting factor, MPF. And again, now you can see that when CDK and cyclins are coming, they are getting conjugated. Now the MPF, which is mitosis promoting factor, it promotes more of the phosphorylation activities of these proteins. And after that, now once the mitosis get completed, the cyclin get degraded, and that process gets complete. So again, it is slightly more complex than what I have simplified in my lecture, but I do not want you to go in that great detail. In case you're interested, this is part from your book. You can read this part as well. So I think one more question was that about explaining a couple of cell cycle based slides again, and I hope I have again taken that for whole class so that everybody is refreshed on those concepts, and those are important thing for you to at least keep in mind, because many of the important life processes, including the disease like cancer, they are heavily regulated with these kind of cell cycle checkpoints. And also the important phenomenon like cloning has also benefited a lot with our learning of how to take these different phase of the cell cycle, how to arrest the cell cycle at the GO phase, and then try to control the cell development in that way. So these are important concepts, but I hope I have already explained that in my beginning slides. Okay, other question is, what about organelle duplication? Uh, how does other organelle, like except mitochondria and plastids, how they self-duplicate? I think that's a nice question. Uh, and so both in the G1 and G2 phase, which is the growth phases of the interphase, right? During those, the cellular organelles, they're also going to get replicated, but not exactly at the as the whole cell organelles, but rather there are some machineries which are making those organelles, right? For example, for the ribosomes, the ribosomal RNA, ribosomal proteins, those being synthesized. For endoplasmic reticulum, there is, different membranous involvement is there, and then cisternae being formed, which gets condensed in the prophase and following the other interphase cycle. Then, of course, you have these self-replicating organelles like mitochondria, chloroplast, and centriole. So, yes, organelle replications is going to also occur during the stages of the cell cycle, and proteins are getting synthesized, which is for different of these organelles. And finally, they're going to put together these machinery and make different organelles in daughter cells. So one question was about the X chromosome inactivation. This is slightly difficult concept to say, and also, of course, I'm not going to ask question in that detail. Nevertheless, since you have asked this question, I have put together this particular stuff with the image to explain. But the key idea here is that uh, the in the female you will have two copies, from the of the X chromosome, when you are going to get the gene products from the father as well as mother. So there is going to be some transcriptional silencing of one set of X chromosome in the female mammalian cell, and that is what is termed in here as a X chromosome inactivation. And again, the concept is quite detailed, when you have to really understand that how the XXXY chromosomes are moving in male and female. In female, with the XS copy, one from the coming from father, one from mother, they are going to duplicate some of these copies of these genes, and therefore that one copy of the X chromosome in each female cell is also inactivated, shown here with this particular cat example of the fur, which we have talked in the CC cat example. So other question, I think the same student has asked another interesting question about gene activation, gene silencing. Although I have explained a little bit some concept here, but I would say that you can wait for these things to come when Professor Rahul Purwar is going to talk about gene expression, gene regulation part. Then these concepts will become much more clearer. But just for your curiosity, I think the gene silencing is going to result from not only just with the DNA alone, but with the interaction of DNA, RNA, protein, and along with some chromatins, which is the key regulators of the gene expression. There are set of RNA known as non-coding RNAs. They also play a very important role in the gene expression. They're also involved in the X chromosome inactivation, and that's where some of these concepts are being taken. Gene silencing and these concepts are also heavily used for the various type of gene therapies. So these fundamental concepts are quite crucial. But again, because this is a question only from one of the student, I'm not going in detail, and it's not going to be within the scope of the exams for any of you. So one more question here is for the retroviral vectors and the Yamanaka transcription factors, which Dr. Shinya Yamanaka, what he described, right? So these vectors, vectors in a crude way is like a car, right? When you are sitting in a car will take you from point A to B. Likewise, these vectors from bacteria or viruses, these vectors, they are like those plasmids, which I probably have explained briefly in the prokaryotic context. So these vectors can be, genetically engineered and modified in such a way that, if you can insert a gene of your interest, and then now this particular backbone of this vector can insert this particular gene of interest, and now it can carry, like the car example which I gave you, from a point A to B, and then make multiple copies of this so that the genes can propagate. This concept is quite well established in the field of gene cloning, which I will talk in my next class when I talk to you about role of biotechnology and some of the key recombinant DNA technologies, how they have emerged. But here, he has used, Dr. Shina Yamanaka used one of the retroviral vectors, which consist of some of the proviral sequences which can accommodate the genes of interest. And now he used some of the transcription factors, which are the genes which are having ability to, getting turning it on. And then they are going to transcribe into mRNA in a particular cell at a particular time. He used some very specialized transcription factors, OCT4, SOX2, MEC, and KLF4, which are highly expressing in the embryonic stem cells. And based on these, then he was able to induce the pluripotency of the cell, which is known as iPS cells. So based on his name and contribution, these factors are known as Yamanaka factors. But I'm not going to go in this level of detail at this stage of your quizzes or your exam. Uh, I think we had talked about circulating tumor cells and how some of the newer technologies are trying to detect those in the human body circulation in plasma and the blood and other biofluids, so that cancer could be detected early. Uh, just to refresh that concept again, I had mentioned, given example like any antibody which can be used to look for an antigen of interest, right? So antigen antibody interaction is very strong. So if you know that there is a specific type of antigens are getting expressed on the circulating tumor cell, you can use some antibodies and which can try to bind selectively to these antigens, and then that could be used to capture those antigens from antibody. So this is known as immunoassay because you are looking at some antigen antibody reactions, and once those have been identified on the cell markers, so again the identification of these markers are coming from the basic science research, coming from the lot of research happening in the field, from genomics, proteomics, other type of cancer research. But once we have identified these markers, then those markers could be utilized to develop antibodies, and different type of detection technologies like CTC detection technologies could emerge. I have given you a couple of examples that time also, that there is cell search system. You can actually Google that and you can find out how that technology works. Very interesting reference, but also some references can be obtained from here, which we have provided this link of this paper. You can read that in detail as well. All right, one more question is the cells shown in the first column have uh, both blue and red, one from mother, one from father. Since these cells are present in our every body cell, so how can we say that red and blue colored paired chromosomes are from mother and father each? What is the homologous pairs? Of course, these are some of the basic question and interesting question which you have asked for. I'd already explained here. Pair of homologous chromosome is one set of chromosome each from the both the parents. So that is from the maternal set as well as the paternal set. That is known as homologous pair definition wise. So here what we are expecting, that the two chromosomes of this homologous pair, they are uh, individual chromosome, but they are going to be inherited from the two parents. And they are, if you look at in the microscope, they are going to look at very similar, but they, for the same gene, they have different alleles, which is again, some of these concepts will be easier when we have talked about more of the gene expression, gene regulation, but I will touch upon this in the genetics context as well. So for the same gene, there could be alternative alleles, which is going to give the corresponding locus on different chromosomes. And that's why these homologous chromosomes which are pairing is very important, although they will look same. So they are not associated with each other in any other obvious way, except that during the meiosis they get paired, and that's where you are going to also see the activities like synapse being formed and the exchange of the genetic material happening, and those are the crucial key points for the diversities of these genes to happen on the crossing over, right? Other question is that the scandal done by Dr. Wong Woo Suk, which was, I talked in the context of cloning of different animals, especially also we talked about embryonic stem cell based work. So yeah, so Dr. Wong Woo Suk, he was really a great scientist to say to, because he really showed a lot of interesting ways of cloning animals. And he, but he started getting so much prestige, so much recognition in South Korea. He became like a really top individual in their country. People started admiring him very much. And on the way, I think, in the wake of reputation and so much prestige, probably he became too greedy, and lot of stuff started coming to from his lab, which was not reproducible. And that's where when he showed about somatic cell nuclear transfer technology, SCNT, those in the science, scientific field, people found that it is not reproducible. A lot of questions being raised, and then many observations came that some of the figures what they have shown in the paper, those were actually not correct. Those were duplication of the same thing in different ways of presentation. Also, some of the acts which they have taken for the experiments were actually not gone through the ethical practices of the approvals. Uh, Even when people have tried to confirm the clones from those derived cell lines, uh, they exactly did not match. A lot of variations were there. So many issues came actually. You can refer to this link which we have provided here. But just to give you a personal perspective to it, I visited actually this same university, South Korea, in 2019, and in the same center where these experiments were happening, I just happened to visit with my proteomics collaborators over there, who is the Korean Human Proteome Organization, Professor Ji Cho, and we discussed a lot of work which we are doing in our area. But on the way, when he showed that this is the building where Dr. Wang Gusok and his team was doing the work, and then in all of a sudden, all those paper which I had seen from Wang Gusok and all those retraction which had happened, just immediately started reminding those, right? And that definitely reminded me that after so many years of those work in 2004 5, still the damage remains in that institute, big university. And that was a sentiment I could see from there. Of course, these are the people not associated with that with anyways, but I'm just giving you some ethical advice again, that make sure that you are thinking about as a part of the big team, that every member has to follow good practices. Otherwise, these kind of blots remains on the university or institution for a long time. So another question is that for the mitosis in the human somatic cell, is the number of chromosome in metaphase is 46 and in anaphase 92? I think to simplify that, we have put together this table, which now you can see that uh, how the chromosome numbers and chromatid numbers are going to vary in different phases, and number of chromosome in the mitosis is going to remain same in all the stages in the cell division. Only DNA content is getting doubled during the cell division. And during the enough phase, two sister chromatids, they are going to separate, which you have seen, which is going to form two daughter chromosomes. And then a similar question. So again, some of these questions we try to club them together. Provide the number of chromosomes in each phase of meiosis. Uh, so again, you can look for both chromosome numbers and chromatid numbers here. Uh, in the interphase or meiosis one, the chromosome number remains same, 46. Chromatids are going to get double here in meiosis one. And after the division has completed, now this number, chromosome number became 23, chromatids became 46. After the one more round, now the mitosis is going to happen. Now that time chromosome number is 23, chromatid number 23. And a question from a student was more like discussion than question, but briefly I'll take it. Common theme for all the cloning product is actually exactly identical to the parent. And I think we just discussed also right now about the X chromosome inactivation. So we totally agree with you that clones are not exactly identical, right? And that's where, despite the success of dolly cloning and many other animal cloning, uh, human cloning and those efforts were totally banned because of many of these issues, right? And but just for your information, because you've asked about this question, that we have put the information here that how the despite that the cloning process is going to be very similar, but there is going to be influence of other type of genes as well, which is the genes from the mitochondria which is coming in the cytoplasm, or even the during the development phase, lot of effects will be also happening from the epigenetic factors, and all of those which might be coming from the uterine environment, even they will result into some differences. So you're absolutely right, they are not exactly identical, although by definition we say that a common theme for the cloning is that the products are going to be identical to the parents. Okay, another question is, does the interphase in meiosis 1 consist of G1S and G2 phases similar to the mitosis? Also, why do we not write a pro-metaphase in meiosis 1? So you are right that the interphase in meiosis 1 also consists of G1S and G2 phase, which will be similar to the mitotic cell division. as I already reminded you that before meiosis 2, there is not going to be another round of interphase, right? So interphase is going to happen only once. So that takes your first query. Pro-metaphase is actually being formed in both meiosis and mitosis, and that is going to help in the all the chromosome and chromatid material to come onto the metaphase plate, and that is going to be common here as well. I think another question, a very interesting observation, in fact, from a student, like, is it possible to perform artificial endosymbiosis and modify a cell to perform more functions? I think you started thinking like a good researcher, which is excellent. Uh, but if you think about the actual, biology and sometimes life science activities versus experimental activity, the time scales are very different, right? So when we talk about endosymbiosis, which is one of its type event of mitochondria and chloroplast, that has happened on the very large time scale of billions of year scale. Whereas if you want to attempt the endosymbiosis in vitro in the lab, then you have to think about many factors, which will require mimicking the environment of Earth condition, the temperature, understanding a lot of other factors which might be influencing that. So a biologist involvement, a chemist involvement, a bioinformatician, system biologic, synthetic biologic, many people's actually team is required to think something along those lines. But I think you are thinking in the right direction. There are other people also who have started exploring these possibilities of at least thinking how to do artificial endosymbiosis and modify the cells, right? And for this type of concept, there are many applications actually when people have started producing more of the biofuels, different type of food production, agriculture, fermentation industries, other type of enzyme production. Many of these things are influenced from it. Of course, nobody has able to mimic what you are trying to ask here, artificial endosymbiosis. But to perform the cell function, there are different applications of that which are similar to this concept, which you can also read in this particular article, which we have provided here. But some of these concepts of how to modify the cell and try to utilize the cell to make more product and perform more function, we will take when we talk about some biotechnology concept in the subsequent lecture. All right, another question. I must say that you guys are doing a fantastic work. Reading really well, watching the video, and reading the textbook, and thinking beyond that, which is I think really a satisfying experience to see at very early stage of your career how sincere you guys are. So, if the gene numbers they do not explain organism complexity, does it signify something else about the organism? So, I had given a concept that different genome sequences from Arabidopsis or C. elegans or Drosophila or human, they were not very different. So, the gene numbers they are not explaining the organism complexity, and so that is the question coming from that idea here. So there are actually, people who have tried to look for the correlation of genome size versus the complexity of an organism, and this particular image probably, will give you more clarity on that point. Essentially, that conveys that even a very simple protist, which is like amoeba type of organism, even they can have very large genome, right? And so the genome of amoeba is almost 200 fold larger than the human genome, which is not expected, right? Otherwise, if you think about the complexity of the organism, uh, so therefore, genome size versus the complexity or the feature or the phenotype, they're not directly correlated. And this particular observation has been studied by a few scientists. They have termed this under C-value paradox, which means that the amount of DNA in a haploid genome, which is one C-value, does not seem to correspond strongly to the complexity of an organism. So therefore, this C value can be very indeterminate, and that is a paradox in this field. A basic question about chromatid and chromosome differences. Of course, we have explained that in different contexts. This chromosome is getting duplicated after the S phase, and after mitosis, getting these now forming these chromatids, which is getting separated as a full chromosomes eventually after mitosis, right? So in the chromosome is essentially the carrier of all the genetic material of the DNA, whereas chromatid actually helps in the process of duplication, and when the division is going to happen, then only you will see these prominent chromatids, chromatin kind of material start getting condensed. uh, Chromosome, they are present in entire life cycle of the cell, whereas chromatids They're only formed when the cell is undergoing either these division of mitosis or meiosis. Again, I have given a couple of points here which you can try to visualize and get more idea. But I hope the key idea of chromatids only appearing apparently in the cell division is probably clear to you now. Does chromosomes in daughter cell become shorter than the parent cells after cell division? So no, chromosomes in the daughter cells are not shorter after the parent cell division, after cell division, because the DNA contents, which is now the sister chromatids, they are going to get separated from each other, and they are going to form the daughter chromosome. So they are going to be exactly same as the mother cells. Um, Another question is whether in the anaphase, how does half the chromosomes or the chromatids behaves as the chromosomes? So we just mentioned that during these cell division process of mitosis and meiosis, these uh, chromosomes gets condensed, now sister chromatids being formed during the process of anaphase. Now with the repulsion of the set of proteins belonging to the cohesin complexes, uh, different now they are getting separases are being used to separate those, and now these chromatids are slowly pulling apart from each other, and they will behave like the new chromosomes. So they are going to develop into the full chromosomes. Please explain this slide of dolly cloning. I'll not go in great detail, but just to refresh you again, we said that from one sheep, the egg cell was taken. From other sheep, the mammary cell was taken. From the mammary cell, nucleus was taken out. And from the egg cell, the nucleus was removed. So now this became enucleated egg cell, which is not having the nucleus. When the two were fused, and for fusing this particular empty egg cell with a nucleus from outside requires a lot of tricks. Could be some sort of electric shock, electroporation methods, or some sort of culture medium, nutrients, which is facilitating this particular nucleus to enter inside the cell. That process is quite important, and that is quite a failure happens here. Hundreds of events only result into one successful of the final embryo to form, and that embryo was actually, after the growth initially in the lab, was transplanted to a surrogate mother, and then the sheep dolly was formed. I think we had some more questions in that, right? I will take in some time. What is pluripotency and totipotency? Uh, a very broad concept here is totipotential, as we talked in the concept of plant cell. They have ability to develop into any kind of cell, and they can be generated into the full plants. In case of animal, human, totipotent cells can give rise to both placenta and the embryo, and the pluripotent cell cannot give rise to placenta. So some of the key differences are mentioned for your interest if you are interested to learn about it, but that we have not talked about the embryonic development, so I'm not going in detail right now about this part. And development we are not talking actually in this course, so it is not required for the course point of view, but for your understanding, please refer to this table. Is meiosis 2 a mitosis of daughter cells obtained in meiosis 1? So I think this particular slide image will clarify this point, and you are right. Meiosis 2 is the exact process of mitosis only, of when now cells are duplicating and two daughter cells being formed. And the point to note is again that there is not going to have an interphase which we see in the mitosis, but here in the meiosis 2, interphase is not going to happen. Why the animal cells need centrosome if plant cell can divide without it? Yeah. So of course, these are very thoughtful research questions. Centrosome is an important part of how the cell organizes the cell division, and the interplay of different microtubules, which is organizing this activity in the centrosome, is required in the animal cell. The plant cells somehow have evolved the mechanism which allows them to divide without the need of centrosome. And again, that could be an interesting research question. I have to now rush. I think I am taking a little longer time to explain each one of these. There's still quite a few questions to actually go through. What leads to disappear the organelles during the prophase? Uh, yeah, so the prophase is the time when cell is actually doing a lot of changes for the division to happen, but these organelles are not totally disappearing, but actually the cell membrane is disappearing at that time. So when you do the staining, it is not visible completely. And along with those, a lot of activities happening in the microtubule and those cytoplasmic elements which are getting organized into this. That's what we have already discussed in the class. Can we put the checkpoint response chemicals from outside to avoid growth of cancer cells? Uh, so cancer is quite complex phenomenon. As we keep talking, you will appreciate how difficult this problem is. But there are different checkpoints in the cell which we talked in the cell cycle, which is quite crucial for if they don't regulate properly, it may result into cancer. And that's where there have been many experiments, many efforts going on in the field to try out different cell cycle checkpoint inhibitors and different chemical like what you thought, whether those could be used to control these cancer cell, right? While this is not completely successful and this is not the only factor responsible for the cancer cell development, but this article, which is a good review article, I've given the reference here. You can use that to read in more detail. But here they have shown different inhibitory drugs at different points of the cell cycle checkpoints, which can be used to control those. And like what you thought, there are other scientists are doing the experiments, which are in different phase of clinical trials, different drug targets of for looking at controlling different cell cycle phases are being tried for different cancer, and their major roles in the cell cycle have been described. So you are thinking in the exactly right direction, and this review article attesting your thoughts and giving you more value to read further. Why did animal eukaryotic cells fail to engulf plastids during evolution? Of course, these are only hypothetical situations which we can think more and more. Uh, Probably, because we can eat, we can take other type of food, so we have resources to generate energy, for life process to happen. Whereas uh, the plant chloroplast, they are going to rely on their food to get synthesized in the process of photosynthesis. They can't just eat the food from outside, the sugar and all. So that therefore, uh, this prediction is that the eukaryotic cell failed to engulf the plastid during evolution, and these are hypothetical possibilities of that. Why did not cell divide after G1 phase only in meiosis, as it would have avoided the metaphase to have two parts M1 and M2? So, again, this part we have talked, again, refresh this concept. Every step is actually very important in the cell cycle and the cell division. While each one of those are performing its crucial activities, nevertheless, the interplay of all that is really orchestrated and they're all involved in the successful cell division to happen. If the cell will divide after G1 phase, then whether it's going to really duplicate its DNA, that is the, going to screw it up. So that you have to keep in mind. For how long can we store stem cells? Uh, we can really store it for long duration, and they usually don't. If you store them in the cryopreserved, with -80, those kind of temperature with the proper preservatives, it will not go bad in the long term. And yes, the parents can also use these, the haplo-identical transplants, where people can use the baby's cord blood as well, which can have almost 50% match. So not 100%, but 50% match. Why the X cell being used, can't the memory cell being cultured as such? If you take only memory cell, you can grow that, and memory cell is one of the body cell which can continue growing. You can maintain that, but you can't make the clone from that, because for when what you are trying to do here, you are trying to take the X cell, and because X cell is only going to make the zygote and develop the embryo. So then this nucleus of somatic cell was taken out and the egg was taken, so that now the embryo could be formed, and that resulted into this type of embryo. For formation of zygote, normal two haploid cells are fused, but exactly what is happening here? Uh, I think, when different sperms, they are reaching here, uh, the pronuclei of the sperm, which is getting dissolved outside and now reaching to the uh, nucleus of the egg nucleus, this is shown here, they get fused together, right? So then this is how the karyogamy happens and the zygote being formed. Are fimbriae and pili same? No, there are some differences. You can see the structure here of fimbriae. These are the pili slightly longer hairs. You can read some of the details mentioned, nicely prepared by one of the TAs, and you can have a look on this part in detail. Why are gametes not formed from gametes? Since then, the number of chromosomes would not have to be halved in the daughter cells. So mitosis generates two new daughter cell, as you know, and the same chromosome number is going to be maintained. If the daughter cells became gamete, the process of fertilization will double the numbers and the entire process is going to get screwed. So therefore, this is not happening. In earlier discussions in lecture 2, it was told that cohesin binds the DNA along with the length of chromatids. There it caused bonding, while here it causes cleavage house. So, of course, that is going to stay together, keeping it together. But then there are enzymes like another set of enzyme, which is part of these complexes, which is enzyme separase that cleaves the cohesin, and then now that can be released in the anaphase. So this was much more bigger complex, and many proteins are actually involved in the even cohesin complex as well. Can we say that meiosis 2 is exactly same like mitosis? Yes. In the third step of meiosis flowchart, we are using the term sister chromatids. I think I've explained that now, but some explanation that we have mentioned here, so you can read that. All right, now let's take a couple of questions on the metabolism. We are getting late, but I will first take the metabolism Q&A, and of course I will come to you, Sai, in some time. So mesophyll cells, they don't contain stomata. They are just present below the layer of mesophyll cells. I think your understanding is correct. These stomata are present below the layer of mesophyll cells on the lower epidermis of dicot plants. Is green color of leaves, what we see in the reflection in the green light. I think, we have already explained that in the earlier concept as well. The red and blue light is absorbed by the leaf. Only green light is reflected, and that's what makes it look green. In the light reaction, I talked about cyclic, non-cyclic processes. Why is emission of oxygen is step 3 and not step 2, since electrons and oxygen are simultaneously produced and later excite these electrons. So here we have talked about the 2 and 3 activities happening. So in step 2, electrons from the photosystem 2, they transmit to the primary acceptor PS 680, sorry. And to fill up that vacant position of electrons, the photolysis of water occurs. So that is the step 3. So these are actually going to happen in that sequence in which the electrons from the water molecules will be transmitted to the photosystem 2. Is photosystem 1, which is the P700, also composed of chlorophyll like P680? So yes, brief answer is yes. There are set of chlorophyll molecules in both the reaction centers of P680 and P700, around 200 to 400 of chlorophyll molecules A and B in photosystem 1, and around 200 chlorophyll molecules of chlorophyll A and B in P680. Accessory pigments like carotenoids are also involved. How exactly do the mentioned processes help in the ATP generation? So we talked about glycolysis. We talked about pyruvate oxidation to form acetyl-CoA, citric acid cycle, and oxidative phosphorylation. The electron transport chain, which is going to synthesize the ATP, it actually works by converting the NADH into ATP as well as the FADH2, and those are going to shuttle through the this process of ATP synthesis, which makes ATP. Again, I will probably refresh some of these concepts shortly in my slide. I will come back to some of you who have raised hand. Let me just go through this. So this comment and a couple of other comments were like that how much of these reactions we are supposed to memorize or understand those in detail. And I totally agree with you that some of these reactions which we talked and different Krebs cycles and steps, those are quite tedious. So that's why I'm now going to give you reviewing some of the important concepts and what is expected out of you to know in detail, right? So please pay attention, then you can, after that you can raise hand. So in case of the photosynthesis and cellular respiration, which we talked, two crucial activities which we discussed. Photosynthesis is helping for the anabolism, whereas the cellular respiration is happening for the catabolism. It means some breakdown is happening and ATP is being generated in the respiration, whereas new organic molecules like sugar are being synthesized in the photosynthesis, right? The summary of the photosynthesis, especially the light and dark reaction we spoke. In the light reaction, which happens in the grana, and the dark reaction, also known as Calvin cycle or the C3 cycle, which happens in the stroma. Just, what I'm trying to convey you here, what is expected out of you to understand in detail, right? So I hope this is just refreshing your concept, and also I'm not going to ask you questions more detailed than what we are talking right now. So from the water, oxygen being released as a part of the activities of the light reactions, ATP and ADP being produced in the light reaction, which is taken by the Calvin cycle, and now the three carbon product which is being formed, 3-phosphoglycerate, forming the glyceraldehyde 3-phosphate. That is why this cycle is known as C3 cycle. And this reaction requires light, known as light reaction. This does not require, so this is the either light independent reaction or dark reaction. So I hope this is now clear to you that the key differences of these two here, CO2 being used and sugar being formed. In the light reaction context, we talked about electron transport systems. We talked about non-cyclic and cyclic chains here. So in the non-cyclic phosphorylation, uh, water is being used and oxygen is released, right? Both the photosystems, photosystem 2 and photosystem 1 is involved. So non-cyclic reaction is linear, moving from here to here, and then even NADPH is being produced, right? So in the process with the cytochrome B complex, here ATP being produced. So key point to keep in mind that in the non-cyclic, which is not making a cycle, it is just moving from photosystem 2, going to the primary acceptor, moving from the cytochrome complexes, coming to photosystem 1 with P700, activating ferridoxin, reaching to NADPH. This is the non-cyclic reaction where oxygen being produced, ATP being produced, NADPH being produced. In case of cyclic photophosphorylation, this part becomes cyclic. So therefore, this part, the left side and the right side, not happens. So only ATP being produced here, and NADPH and oxygen is not going to produce. I hope this just makes it very simple for you. Now the Calvin cycle, I already briefed you that the 3-carbon sugar, glyceraldehyde 3-phosphate, is the most important entity over there, which is, you have to understand why we name Calvin cycle the C3 cycle. I'm not going to ask you questions about the intermediates of the Calvin cycle. So that is just for you to understand the steps, but not essentially to ask you all the enzymes and the reactions of that. Now, in the C4 cycle and the crassulacean acid metabolism, which we talked about, the plants which are living in the arid conditions, which is not normal, it's more diluted conditions. So in those, the C4 plants, why we say them C4? Because the first stable compound being formed, that is known as oxaloacetic acid, or OAA. And so you have to keep these differences in mind, that from the uh, C3 cycle versus C4 cycle, what is the major products being formed, what is the major differences over there. And then in the C4 cycle, we are looking at, along with mesophyll cell, we also have bundle sheath cells. And then in the CAM cycle, castration acid metabolism, we have similar phenomenon occurs, and that's why they are trying to preserve the water. They are living in the more desert conditions. So stomata remains closed in the daytime and open during the night time. Many of the activities are actually governed with the enzyme like PEP carboxylase. Uh, just the overview of the cellular respiration, the key steps we talked, from glycolysis to pyruvate oxidation, citric acid cycle, oxidative phosphorylation. In the glycolysis, what you should be paying attention to, that one glucose molecule, when the input is glucose, after end of glycolysis, two pyruvates are being formed, two molecules of ATPs are being released, two molecules of NADH being released. Of course, there are 10 steps of these reactions. I would not ask you questions about what each of those steps are, right? You have to keep in mind there are some steps which are energy investment phase, when the cell is actually spending ATP for activities to happen. Few are the energy payoff phase, when ATP being produced with the substrate level phosphorylation. In after these, from glucose to pyruvate being formed, now pyruvate is getting converted to the acyl coenzyme A, and that is the crucial activity, moving from cytosol to mitochondria. And during the process, NADH is again released. Now, from this particular process of pyruvate, now this particular pyruvate is entering in the mitochondria and oxidized to the acyl CoA, which is further oxidized in the TCA cycle or citric acid cycle, which is involving series of reactions here. Again, expectation is not for you to know all those reactions, but rather the byproducts which are being formed. How many NADH molecules, how many FADH molecules being produced, how many ATP being produced. And this process we talked about the ATP synthesis. Here, NADH requires involvement of three complexes, 1, 3, and 4, whereas FADH2 is requiring complexes only 3 and 4. And while in the NADH, 3 ATP is produced, in the FADH2, 2 ATP being produced. So overall, the expectations are not that you should be keep remembering all the enzyme steps, but crucial byproducts which is generated in each of these steps, and also then that is going to release what number of ATPs in those processes, right? So two ATPs as the process of in the glycolysis substrate level phosphorylation. Here again 2 ATP. So total we got 4 ATP at the substrate level phosphorylation, and with the oxidative phosphorylation, about 26 or 28 ATP being produced, depending on which type of transporter electrons they are being used. And that is slightly more of a research question, so I have not gone in detail about that. Total 30 or 32 ATP being produced. One of the tea has produced this very nice table, which I thought to share with you. And this will really help you to understand different steps of glycolysis to Krebs cycles, a number of ATPs and their, total, how much being used and how much being produced. That's how these numbers are coming. And an estimate is that one NADH molecule generates 2.5 molecule of ATP, and one FADH2 is giving rise to 1.5 ATP. So depending on number of NADH and FADH2, which we are going to count, those can be calculated with 2.5 or 1.5 to generate these numbers. But I will try to keep these questions very simple. So don't get too much afraid about those. Very briefly, in next 1-2 minutes, I will take a few questions which some of you have raised for the quiz as well of your batch. Uh, so this question was straightforward when we had asked for in the first quiz about the base pairs of AT-GC. So you know that AT and GC going to get base pair, and this is how the numbers came from that. Other question was to do a simple calculation of length of the DNA when you had to calculate what is the distance of the nucleotide base pairs and total how many base pairs we are giving, 6 billion. So that was a simple calculation to give around 2 meters length. Tree of life, which three domains are more closely related? Archaea and eukaryotes, which we have talked a couple of times earlier. In nucleus, most of the genes are there from DNA, RNA, and histones. Lysosomes are not there. Lysosome enzymes are different organelle which are present. In case of gram staining, we had different steps we talked, from the crystal violet to washing it off with the decolorizer alcohol wash, then adding the safranin as a secondary counter stain. Those steps are mentioned here. Endomembranous system, when we say, that involves series of membranes from the endoplasmic reticulum of smooth or rough endoplasmic reticulum and Golgi apparatus. We asked for the bio-inspired engineering examples, and the evologics was actually example for tsunami alert system of what we learned from dolphins, not from the beetle. We had discussed about microscopy, and the differential interference contrast microscopy will have the dead cells. Few queries came from this side as well, that about the gram staining, which we have spoken, that the role of peptidoglycan and lipopolysaccharide membranes. So again, in the gram positive versus gram negative, as we have seen in the structure as well, in the gram negative, we have very thin peptidoglycan, and also we have additional outer membrane, which consists of lipopolysaccharides. So now in the staining procedure, till primary stain, things are happening uniform for both of them. But after that, once you are adding the decolorizer or the alcohol, that actually breaks the outer lipopolysaccharide membranes of the gram negative bacteria, and therefore it is going to start differentiating at that time. And then of course you're going to add the crystal violet, which is washed off from the gram negative, and therefore both the lipopolysaccharides and peptidoglycans are playing important role of differential staining, what we see in gram positive and gram negative bacteria. Which of these organelles is involved in the breakdown of fatty acid molecule? Straightforward answer is peroxisome. All right, it was a tough, long one. Okay, so I hope I am clear with these questions. These questions, some of you have actually requested to share these questions with you. So the entire questions and the possible answers is all going to be recorded, which I'm going to share with you in the recording. So you will have access to everything, whatever we have, right? Whatever questions I took are only from your batch of D1, D2 batch. And likewise, I have many interesting queries from D3, D4 batch as well. And we had a different quiz for them. I'm going to upload a new video after my afternoon lecture to them. It will be a good idea, advice to you guys to also watch other video of the live interaction of the D3, D4 batch also, so that you will have access to more question bank, more concepts, more things, so that it can be for both the batches. So what is the time now? I have exceeded the time 1 hour. Uh, okay. So Ankit, are we ready for the quiz to start?
Yes, sir.
We are going to do on the safe, or we are going to do on the
So we are going with the Google form only. So
uh,
Ayushi will be sharing the link. So
okay.
Yes,
So
sir. So, sir
1 minute.
Ayushi here.
1 minute, sorry, Aishi. So to
Yeah.
all the students, we have talked about two different topics. One is about the cell cycle, one is about metabolism part. Those students, if any one of you here who have raised the hand, if you have any question about the cell cycle part, which is going to come shortly in the quiz, I can take that right away, right now, because so that your curiosity can be, we can discuss that right now. If you have questions linked to metabolism, please put down your hand right now, so that we can discuss that after your quiz. Right now, I can still take any question if it is linked to cell cycle part, which we have spoken. So only keep those hands raised which are linked to the cell cycle part. So let me ask Sai Pavan to ask his question.
Sir,
Yes,
I have a doubt in the cloning part.
all right.
We are removing that egg in place of dolly. We removed that egg nose. We removed the nucleus of the egg nose, sir.
Sorry, your voice is breaking. Can you repeat the question? So in the case of Dolly, we removed the egg from one sheep, yes, and that nucleus was
We
removed.
removed the nucleus of the egg nose, sir, and made
Yes,
an enucleated egg nose.
correct.
Why are we doing that? Can't we mix them directly?
See, so if nucleus is already there, then how you are saying, so what you are trying to do, the normal fertilization process will involve the ovum and the sperm to fuse, right?
Yes, sir.
What
Why
you are
can't
trying to say that without need for having a sperm or a nucleus coming from a sperm, or without having a father, you are able to make a full child, right? So in that way, now you are taking only the female part and the ova from that, removing the nucleus from that, and now taking any normal mammary cell and taking the nucleus of that, trying to transplant that. So that now what you are saying, you are trying to make the clone of that. You are not doing the normal fertilization process here. Otherwise, the test tube babies and many other concepts are linked to that.
like, if we remove the nucleus, then it is called cloning. If not, then it will become like test tube baby like that. It will change everything.
Correct. So cloning is because you are taking any other normal body cell, which you are like from the memory cell or the skin cell or any other cell, which you are trying to take their genetic material and putting that in the new ova environment, and then trying to make multiple copies of that. So you are in some way only utilizing the 50% of biological material, right?
Hmm. Now,
All right, Hardik, you have any question?
sir, one last doubt.
Yes. Sorry,
If
yeah, go ahead.
for normal cell, if we expose UV light, they won't lose nucleus.
For normal cell, if we
Expose UV light.
well, extra UV light is very harmful. It is going to degrade. Lot of cancer also linked to the UV light, right? UV radiation is increasing in general, but also with anything, if you get UV exposure, that is going to be harmful. So even a normal cell, when you do extra UV exposure, it will get nucleus will get degraded.
Okay, sir.
All right, Hardik.
Sir, how do plant cells replicate without centrosomes? This question was in the chat.
I think your question was there, and we don't have a right answer to that. I mentioned that in the animal cell, you don't have that capability of doing it on their own. That's where the need for microtubule and cytoskeletal elements are there. Plants still have the capability to do that automatically, so they did not evolve these part. But again, there was no direct answer to this question, so that's an interesting question. Other student, Utkarsh.
Sir, is there any specific breed of the sheep that were taken in the Dolly experiment?
Any specific need for?
Breed.
Breed. Okay, no. I'm hoping it was not, but probably they would have taken some breed of good commercial value, right? Because in the sheep you have a lot of wool and those things, and that is one of the concern that are we going to make the factories like animals to produce these type of products, right? But I'm not sure for the Dolly experiment, was there any specific rationale of choosing a breed?
Okay.
All right, yes. Yes, please go ahead.
I don't have. I'm
All
not.
right. All right. So any curiosity question we can take later on if required. If I don't want to have the whole class staying here. Uh, any of you who are curious for any other specific observation, we can take that. Now let's start the quiz. I hope we have already gone through the contents. You're all refreshed and pretty much well prepared. Questions are not made to penalize you, just to be very clear. Questions are actually meant to just make sure that you are reading, you're understanding, and we are not going to the to the stage when we want to penalize you for the marks. So quizzes are being made quite moderate and more straightforward. All right. Okay. Good luck for the quiz two to start, and I